It is in process. So I come back here. I start again. That's it. And here we are. We start. Good afternoon, everybody. We are going to wait a few minutes until everybody is logged in. I see the numbers growing up. The good news for those who are logged in first, we are going to be more than 100 this afternoon. Okay, 62. Now it's stable at 65. I will start with that. Welcome, everybody. Good afternoon, good morning for those far away. This is the first Committee of Expertise for Work Package 1, UNICOM Work Package 1 this year. And I'm sure you will be highly interested in this presentations and discussions. The topic we're addressing is vaccine challenges. That was an echo from, sorry for that, from YouTube. We are talking this afternoon about vaccine challenges, cleansing, confidentiality, and vaccine naming. And we have three speakers with us this afternoon. I will present you these in a few minutes. As usually, and for those new with us, we have some rules for this virtual meeting. The first thing is everybody is on mute. You can interact with the speaker and the panelist by using the Q&A facility, which I'm going to show you. And you can possibly sometimes also speak, and I will give you the, as a participant, I will give you the microphone for that. But in that case, I ask you to be concise and let's enough space for a conversation and answer to your question. On the Q&A facility, you can also show your approval. That's a way also to change the sorting of the questions. And also a very important point is that after the meeting, you will have a questionnaire to answer, which will be sent to you possibly tomorrow, so 24 hours after this call. How to access the Q&A facility? You move your screen on the window, and you should see the toolbar appearing on the lower level. And then by clicking on the Q&A, you see something like this, where you can ask a question. You see here, the question starts with, "Dear Christian, can I ask you a question?" And then I answer that. You will see the answer, or respectively the person who answers you, see the answer, and you may comment. By clicking on the thumb up, every participant can, as I said, modify the sorting of pending questions so that we have important or interesting questions on their point of view first ranked. Security. We have 2 years experience already with Zoom. Security is our priority. It's also Zoom's priority, actually. And when you register to this session, you have a personal link to it, and it is, for the rest, all password protected. So we expect that you don't share your personal link with anybody else. If another person would be interested, that person can register. We have enough space for that. The session is recorded, It is made available on YouTube channel. It is already broadcasted now. And if we would have people not able to access via Zoom for possibly company reasons, they can access directly on YouTube. You will have a questionnaire to be filled, as I mentioned, but we will say that again. Who are the speakers today? We have ladies first, Annet Rozema, who is working at CBG in Netherlands. She's also leading one work package at UNICOM, and she addresses substances and cleansing, for instance. We have Olof Lagerlund. He is based in Sweden. He must be Swedish. And he is also engaged in work package 1 in UNICOM. Not only work package 1, also in substance cleansing from its job at UMC, so Uppsala Monitoring Center, the WHO competence center there. The third speaker, Jean-Gonzague Fontaine, is a colleague from ISO TC215 working group 6, but his main job is at GSK Vaccine, where he has some responsibilities regarding substances, among others, in the IDMP context, of course. On the panel, we have two ladies. First, we welcome Rafaella Balocco. Rafaella is working at WHO headquarters, and she is responsible for the INN program, among other responsibilities. Then we have Malin Fladvad from WHO-UMC, so the same place as Olof Lagerlund, and she's in our panel to participate to these discussions with the others. So that's the panorama for us this afternoon. And now I'm going to hand over to Olof Lagerlund, who will start with us, and then the conversation will continue with Annet Rozema and close with Jean-Gonzague Fontaine before we have the discussion. Olof?
Yes, thank you. Good afternoon, everyone. I'll try to give you a global perspective on pharmacovigilance and vaccines a bit from our part. And you can, next slide. A short, brief outline. I'll talk about the pharmacovigilance and the UMC perspective, and then also a bit about the global PHPRDs and the global vaccine initiative, and then some final remarks, and then be happy to take questions afterwards. We can take the next slide. I don't know how many of you that know, but what Uppsala Monitoring Center is and what we work mainly with is the WHO program for international drug monitoring. And this was created in 1960s as an aftermath to the discovery that thalidomide could cause limb deformities in babies. And when it was prescribed for morning sickness in pregnant women. And the need for systematic collection of adverse event information, both during drug development and also after post-marketing, became apparent. And it started out quite small. I think it was about 10 members that started to sponsor Uppsala Monitoring Center in '78, but now there's more than 170 full members across the world, covering most of the world's population. And as you can see, Uppsala Monitoring Center has been responsible for the technical and operation aspects since 1978. And I won't go into everything about the program or UMC, but I will mention Videobase because that's the global database of ICSRs, the spontaneous reporting from here there. And all members in the program are encouraged to share reports to Videobase so that you could have the detect something, even if it's rare occurrence in different countries all over the world, since a lot of the industry today is global. And we use different things to structure these things when they come in, like Medra and WHO drug. You can switch next slide, please. As you could see on the previous slide, we ran out of buildings to compare the stack of all the ICSR that are in Videobase. And the COVID-19 vaccines are contributing a lot to that. You can see here on the slide, on the top, the all vaccines, it's nearly 3 million ICSRs now. And you could compare that to the influenza vaccines, for instance, that has been around for a number of years, that we have like almost 300,000 reports on, or I should say only 300,000, so 1/10. So it's really getting a lot in here. And the most common ICSR for the COVIDs, Astra, Community, Modernas, you see, you don't really enjoy the terms if you're like into the naming. So that's one part that we will discuss today. But it's also, I just want to mention that the vaccines here are the ones that are the most used in the countries that share the most reports. And the top five is US, UK, Netherlands, Germany, and France that share the most reports into Videobase. And then it also gets back that maybe the Sputnik or the inactivated Chinese vaccines aren't that well represented here. We can take the next slide, please. Here is a slide I put together with, since it's so many reports, you can imagine many different ways that the vaccines are actually reported. Here are some examples where we have the trade names in white, substances in bluish black, and then we have sort of nicknames, or what to call them, in brown on the bottom for the inactivated ones. And these may not seem like, but there have been a lot of issues with the trade names, what to call them. And especially this was true for the first couple of vaccines when there were emergency use in a lot of countries, because then they didn't have proper trade names, what to call them, how to report them, what is the substance in them, and so on, how to differentiate one substance between another, one vaccine from another. And all of this is we try to map, and we do that as best we can. But you can see, like on the bottom right, the Covilio in the activated vaccines, they have the same trade name, but they are composed with different strains from the same EMA holder. So it can be quite tricky, and it's still quite tricky in a number of countries around the world where you can have rather limited information on that. One thing that's very good is that INN, and maybe Rafaella can tell you more about that later on, have named at least the mRNA vaccines, the Tocilizumab, Elotuzumab here. And that makes it a lot easier, especially in combinations with that they actually use the same global trade name for most of them. Next one, please. But apart from naming, there are other regional differences that can be maybe not all noticed on the ICSR, but that we are aware of. Here's one example where you have the dose form differed. I think the UK now has changed to also have dispersion for injection, but it just goes to show that it's not that easy unless you agree beforehand. Otherwise, you have to amend afterwards, and it's not the whole thing. Next slide. Same goes actually for another thing like strength. Different units results in different strength, and this is also might be a problem. Next slide. So why do I talk about this with the differences and and the pharmacovigilance? Because that's one of use cases for the IDMP, and here you can see where we will have it implemented in the when we're shifting from the E2B-R2 format to the E2B-R3, where we, instead of drug name and substance, will use or could use the MPID, the PHPID, the product name part, substance ID, substance ID instead of name, for instance. But as you also can see on the slide, so is it that each ICH region is to implement their own guidelines for ICNMP implementation. Hopefully, we would hope that this was, but that we could have it more globally harmonized, of course, since we are on a global work on a global base. Next slide, please. So there is a risk of local assignment for this. If you have it locally, the assignment for the substance ID or the dose form or whatever is locally, then it would, or local PHPIDs, then that would be sort of, you have to map them later on, and that could be a lot of work. But this slide is only to show you and remind you that I guess most of you have seen what the PHPID is built of, have 4 levels, and that it's the substance, strength, and dose form in some different combinations, the different levels. And we would, of course, like the PHPID level 4 to be used on all reports. But we also know that that it's not going to happen, because a lot of the vaccine reported terms aren't as good as the ones I showed. It's just COVID jab or AstraZeneca misspelled in 5 different ways, and hopefully vaccine afterwards. And if you go into the report, maybe you can find out that it's the COVID or not some other vaccine. Next slide, please. So we are, UMC has been, are going to have the global PHPIDs. And the good advantage with that would be that it would be a single database with having a global PHPID compared to each region creating their own local ones. And it would be a global validated one. We would also limit unnecessary data. We would also have transparent business rules, so it would be easy to follow for both regulatory authorities as well as industry, to see what is it that makes up this PHPID, and should we apply for a new one, for instance. But we also know that the global substance ID is key for all these, because that's part of all the different levels of the PHPID. And if we're going to have a global PHPID, the level 3 and 4, we need to have consensus on the dose form IDs. That goes also for the strength, that we need consensus on that, but that's a little bit easier, I would guess, for most of the things. But how to achieve this? On a global, and a small part was that we started last year a collaboration between UMC, US FDA, NCATS, and the EU ESRS project to have the global vaccine initiative to investigate the feasibility of having global substance management with regards to technical aspects on a global level and scientific aspects as well. Next slide. And here are some things that we discussed and investigated during the year. And we had some performance tests with the exchanging non-confidential vaccines between the NCATS public GSRS and the UMC SRS to see if they could be transferred between each other. The systems could talk in an easy way. And then we, of course, user requirement, we discussed search functions, how to know that we had it in there and so on. But the main important thing there was probably that the vaccine are complex. And there was a lot of feedback for the system development team how these, because it's not just one field transfer, like one structure, but it can be different layers with alternative definitions, protein definitions inside that vaccine, how it's connected to the adjuvant, and etc. Industry was is expressed an interest in joining the discussions, of course, especially with regards to data exchange. And you'll probably hear more about that later from Jean-Gonzague. We managed also to create technical wise substance IDs and PHP IDs generated based on these vaccines. I'll show that a bit later as well. Next slide. We have an agreement hierarchy that I will show you in a minute. We had a lot of discussions on naming, and is still, of course, going on. But one of the main things we did in this project was that we compared vaccines in both the US and the EU to see what are the differences. Are there any differences? And we could find differences between how they were entered in the system. And there were no showstoppers, nothing big. But of course, that's one of the next steps, that we really need to look into these small differences, see if they are something we have to align in a global way, or if we can just let it be and have it as a regional difference. When to draw the line and how picky should it be. Also, this discussion of minimal fields, what do we need to make sure it's a unique substance, especially when with regards to that we don't have non-confidential data in the UMC ESRS as it looks. So that can be a bit tricky then, because some of the data that makes it a unique substance may be found in confidential data. And there's an aim for achieving the global agreement of the naming, so we actually have something global that we can work on together. Next slide. Here is the hierarchy I was talking about a bit. And this is a bit because I told you that we get like reports in that says COVID jab or Pfizer vaccine and or similar. That's not very specific. And maybe it's not specific in a trade name wise or a substance name. So we need some sort of concept or grouper that can have them like on the organism, or a high level vaccine substance that then can be further broken down into more detailed substances, for instance, as you will probably see in Annette's presentation a bit more. And then we will have adjuvants in there as SSG1 connected to the vaccines, and constituents. So what if you take the Corminati, for instance, tocilizumab is when you get the actual vaccine, it's in a lipid, and the lipid is the adjuvant, and then the constituents of that lipid should be also there connected, so you can have a lot of information and follow what the side effects might be later on. Next slide, please. And this is a possible global vaccine process. And it's just a scheme. It's nothing decided, so don't worry. What we talked about is the EU ESRS and GSRS, that's the US, and then it could be any MCA, any authority across the globe that has to market vaccines, that exports the non-confidential data, and it's validated before going into UMC ESRS to check if it's a duplicator is in there somewhere where if it's correct. And hopefully it will be easy and assign a global unique ID without any issues. But if there are issues, then we would have some sort of expert team that could discuss these and how we should assign what we should do. And hopefully the same, you can write more business rules when you get more tricky examples But then, of course, it's probably going to be new types of vaccines coming along as well. So we will probably have to have some issues quite along the way. And then in the UMC ESRS, you could link it. You could have the different codes like the Unicode or EU's TCT code or any other national code to that substance or a cost number or something like that. I'll put the industry on the right left top corner, because they are going to provide all the data from the beginning. They are the most crucial in that sense. But and right now, I think it's drawn like the industry would first get to the authority and then the authority to us, but that's not decided either. It could be that the industry actually give us input directly as well. But for now, I think it's we will see what happens there. Next slide. And this is just showing that using this and the approach we had and the expert team, then being the people in the Global Vaccine Initiative that we discussed this a bit with, we agreed on which strength should we use, which form should we use for the COVID-19 vaccines. But we were able then, and what substances, and to produce PHPIDs for the vaccines by using the ISO standards. Next slide. So hopefully this, with all these different names and substances that have been getting in there, if we would have a global substance ID and a PHPID, maybe we could avoid at least some of the differences here and make it a lot easier to map the reports and get them into the system, and then the analysis of the reports would be easier and better. And of course, like the INN is very great, but when you don't have it, and there are maybe then we can get them to use the PHPID instead, for instance. Next one. And some final remarks. There is a need to work on the global scale, as we see it, to get away from this all different codes. Of course, you could have the PHPIDs on local scale, but then you would have to map them again. And it's better. So, and we see it as feasible to work like this. The more people involved, the more countries involved, of course, it's going to be more of that, but just set up the structure. And the global substance IDs and PHPIDs can be assigned to vaccines and therefore can help in improve pharmacovigilance, especially when we have it. It's been quite a mess sometimes. But we also need umbrella group terms for vaccines, like just COVID vaccines or mRNA COVID vaccines, since the reporting that we have seen isn't that specific. And then final next slide, questions in the Q&A facility. And yeah, I'll be happy. I'll leave over to Annet. And
thanks, Olof. That was an interesting presentation, so thanks a lot. I will touch upon the same topic from the angle of ESRS. As you know, we are work package 2 of the UNICOM program. I present this on behalf of the experts in the ESRS team. I'm myself not an expert, but a project leader for ESRS. So nice, thanks to allow me to present on behalf of the team. Next slide, please. These are the topics that I will address. Next slide. And the topic for today was vaccine challenges with regard to cleansing, confidentiality, and vaccine naming. And I must say that these are all relevant to the substance experts working on vaccines in the ESRS implementation project. So I'm happy to talk about this. Next slide, please. Yeah, about the ESRS project. Next slide. Yeah. So Just a recap. The EOSRS project is making use of the open source software GSRS, Global Substance Registration System, developed by the FDA, and that is developed in line with ISO IDMP. So that means that it describes substances in a scientifically sound manner and with substance class specific data capturing. And in Europe, we have, of course, also as part of SPORE, the SMS database, which is the substance management system, but that is focusing mainly on names and codes. And the difference with GSRS software is that there you have substance class specific details that really characterize a substance. So the key components of the project is cleansing the European data from SMS, so the names, the preferred terms and aliases from the SMS data, but then also building substances in EOSRS as full records, describing the necessary details for a given substance class. We perform the software installation and validation. It will be handed over to EMA later this year. We establish a substance validation group with substance experts from across Europe. I think by now we have about 12 agencies joining the SVG. And very important, the last bullet is that we also make sure we have guidance documentation and procedures available to make sure we have a long-lasting good quality in substance management. Next slide, please. This is a slide that you may have seen before. It is created by Tyler Perry from FDA, who we work very closely together with. I always find this a nice picture because it shows the difference per substance type in what defines that substance. So where it's a chemical, you see that the chemical structure is the defining factor. But if you have a vaccine, maybe you can press one more time. Okay, press one more time. Yeah, thanks. For vaccines, vaccine antigens, these are among the most complex substances to define. The description starts from the pathogen defined as structurally diverse on the right-hand side of the screen, and then specific antigens can be described, for example, as proteins or polymers, and inactivation of toxins and absorbing aluminum are described as specified substance group 1. So you need different components and different types of substance types to define one vaccine, which is in itself already showing how complex that is. Next slide, please. So why is the description of vaccine so important? Regulatory assessors, they have benefits from gaining insight in the scientific description of the vaccines, the relationships, the references, identifiers. I saw a question in the chat earlier about different instances and different codes. We will have in EOSRS not only the EU identifier, but of course also the UNI from the US, and hopefully in the future also the global substance ID. There's different names, so you can search based on preferred term, but also on aliases. There's hierarchy, and it's also easy to access information of similar vaccines. So that it will be for Europe the single point of truth for vaccines, and of course maintained by a shared effort. And the experience from experts who are using the system now during the project phase is that vaccines described through structured data in the SRS system explain so much more than many words. So we look forward to build the full set of records. Next slide, please. Yeah, so what about cleansing of SMS and building vaccine substances in EOSRS? I think that is good to explain today. Next slide, please. So this is a graphic to show you how we work. So there are vaccine data records in SMS, meaning a name and a code. This list is extracted in an Excel format and provided to the substance validation group. They identify if there are any duplicate vaccine substances, invalid substances, and confirm vaccine names. So what is the preferred term and what are possible aliases? And then apart from that, they build vaccine records in EUSRS with all the necessary information in accordance with ISO IDMP. Next slide. So what are we talking about? It's about 550 unique human vaccine substances that we have received from SMS. We group them in by microorganism, and that is about 42% are viruses and 58% are bacteria. And each microorganism contains then of one or more vaccine records and one or more names, so preferred terms and aliases. But the SVG, they process then the records per microorganism. That means that per microorganism, the naming rules are agreed. The review is performed of the SMS records, so the names of SMS. Feedback will be provided to the SMS team, and then an example record is built in EUSRS, and a description is made. And if that is done, it is that's the start of building all the records for that particular microorganism in EUSRS. Next slide, please. So some facts and figures. Not surprisingly, the influenza virus is has the most vaccines available in SMS. So 172. This is the top 10 or so microorganisms. Probably COVID vaccines will be soon also on this list of the top 10. At this moment, 75% of SMS records are processed by mainly by Herman Diederik in support with by other vaccine experts, but he is making the start and doing the bulk of the work. And maybe good to mention here is that many of you know that we also have cleansed the SMS names for chemicals. More than half of the chemicals did not need to be changed in any way in SMS database. That picture looks pretty different if we talk about vaccines. So there's many more changes needed to the SMS data, and we will discuss with the SMS team in the upcoming period how to approach that. Next slide, please. Yeah, so there are challenges with cleansing. And that has to do with the difference of the data available in SMS versus EUSRS. So if you click maybe one more time. Yeah, so in SMS, there's limited information per substance. So it's names, preferred terms, aliases. There it's also translations. Identifies, some of them, not many, I think. SMS ID, XEVMPD code. But then if you look in EUSRS, there's much more information necessary. Source materials, fractions, modifications, relationships, this data is not available in SMS. So the SVG has an issue there, because how to get this information? Maybe you can press one more time. Yeah. So the question is where to find the information, and that is taking a lot of time. I think if you press one more time, Christian, yeah. So building the records in EUSRS may not be too easy. It's certainly not straightforward, but it is doable. But the most time spent, what we see is trying to find the information, the missing information to build the existing substances. This is very much specific to the building vaccines of existing records of SMS. So in the future, we will need to draft a new process, and then we can make sure that the SVG that we discuss with industry and with the SMS team, how the SVG receives immediately the necessary information to build the records in EUSRS. But so far, for the legacy data, yeah, we do not have that available. So that is a worry, and that is also the area where we received the offer from industry to work with us together to make sure that there is less time needed for finding information. Next slide. Okay, then vaccine naming and structure. Next slide. This is a complicated slide, and I will try not to spend too much time because Olof also touched upon it, but it is good to talk you through. So can you press, yeah. So the top level is the organism author level, serovar, strain and fraction, strain and conjugated fraction and adjuvant. And then there is some examples for a bacterium, and I will not name it all, but you can see there is a lot of discussion that went into the syntax for each of those. So maybe you can press a couple of times. Yeah, thanks. So this is the example is for the Neisseria vaccine. And that is also one of the vaccines that we used in the Global Vaccine Initiative to see if we could align between US, Europe, and then see how that would work out with the UMC. So that was useful. There was a lot of information already built in EUSRS. Next slide, please. This is, I have a couple of slides with some screenshots directly from the EUSRS system. So here you can nicely see the hierarchy of the Neisseria, and there you see the serogroups and the strains, how that builds up into the system. And you can imagine that if you are an assessor and you have this so clearly specified, and you can click on it and look into the details, that it really supports you in your work to assess the dossier. Next slide, please. Yeah, again, some screenshots. On the right top of the screen, you see starting materials, repeat units, end groups, and maybe another. Next slide. And here you see some other components. On the right bottom corner, you see the subunits for the Cron 197. So the bottom line here is there is much scientifically sound information captured for each of the vaccines in EUSRS, and we now have the huge task to make sure we build it for the existing vaccines, and then also make sure that we document how we do it from now on, how we build records moving forward. Any alignment that we can make between US and Europe, and possibly also UMC, is helpful, because if we in the future then would want to grow to global substance management, then the more we are aligned now, the better it is, of course. Next slide, please. So there are some challenges with confidentiality of names. And that is related, and I think Olof also mentioned it, it's related to the strain name. So and the fact is that we would then say, if you scientifically, from a naming perspective, look at the name of a vaccine, the strain name, and that was also visible in one of the screenshots, the strain name is part of your preferred term, right? But in SMS, the preferred term is always public, and therefore the preferred term may be in the SMS database, but not classified, not ticked or marked as being preferred term, because it should be hidden from the public data set. In EUSRS, that system will only be accessible for the time being to EU regulators, and therefore the confidentiality of names is less of an issue. There are solutions, and we need to, we are now at the point that we say, okay, we need to sit down and see what are the options, because you want to have the SMS and EUSRS systems in sync, also in the future where there's an interface. So there needs to be a solution drafted to this topic. Next slide, please. Collaboration. So next slide, please. So as you probably know, there are the team, the EUSRS team is quite large. We have, which is good and nice. We have representation of 12 agencies across Europe, and also UMC and FDA are. Olof is one of our SVG members, and we have a very close collaboration with the FDA team. The members are both actually, good to mention, from human as well as veterinary side, so that is also helpful. If you click one more, so the veterinary, so there's different expertise available in different agencies, of course, and the agencies from Spain and the Dutch agency, and Olof is joining from the Uppsala Monitoring Center. And EMA, there are the vaccine experts working together on the vaccine data, and together with FDA as well, We have worked on the Global Vaccines Initiative last year, and there we also reached out to industry, and they reached out to us, and the intention is to work more closely together. So it's a very good, I think, joint effort what we're working on. Next slide, please. And this is, I will touch upon this shortly. The idea is at this moment for vaccines is that at industry side, they will have their industry SRS. We will export from ESRS the substance data, which is called a JSON file. We will submit that to industry. They will import it, edit the vaccine with the information that they have on their end, and send it back to the SVG. And then SVG receives hopefully a solid vaccine record, and will be able to validate the records without having to look for all kinds of documentation and data. So that's the hope. That's the what we are going to work on in the near future. Next slide. So the EUSRS Steering Committee endorsed the involvement of industry in the building of vaccines in EUSRS for legacy or existing vaccines. Whether this would be a future model of exchanging data cannot be confirmed at this stage. These things take time, and three pharmaceutical companies have indicated interest to join this activity, so we will work with them together. And this fits with also the Global Vaccines Initiative, and we expect that we will get also some feedback during this joint effort, that we will receive input for the GSRS software. And the intention is to kick this off early February and confirm next steps. Next slide. This is my last slide. This is what we hope for to have as a future model. Olof also shared a possible view of how to work together. This could also be a way. Suppose industry would have an SRS or similar system. If they would then submit records to either EMA or FDA, including the confidential data, we would validate, confirm that yes, this is correct, and then it would then be up to industry to also share it with, for example, with FDA, and the non-confidential information with WHO-UMC. So that would be that is what we hope for, what is possible. But there is quite a way to go. But this is a possible model. So that's it from my side. So I will hand over to Jean-Gonzague.
Thank you. Thank you, Annet. Jean-Gonzague Fontaine, so I'm part of GSK in the vaccine business unit, and so I will bring here the industry perspective on this substance topic and pharmacovigilance consequences, but also PHP ID. Next slide, please. So just to be clear at the beginning, I'm not binding anyone by what I'm saying. I'm just myself with my expertise. I'm not going to bind GSK in any way or industry in any way in what I'm going to say. It's really thinking prospectively. Next slide, please. So that's what we are going to discuss today. The fact that IDMP for the pharma industry is quite a paradigm shift. That will be the first aspect. Then this implementation of IDMP, so with a focus on substance, what are the challenges and the benefits, and then how we contribute. Annet already gave some direction on that, and I hope I will be able to answer to some maybe questions that were raised by Olof in the initial aspect, in the initial part of the talk, and by Annet on how industry can contribute to this effort. So next slide, please. Let's start with the paradigm shift for the industry. So pharma industry and regulatory process historically are very document-based, very much relying on electronic papers or PDF documents. So except CDISC, which is there since some years now, it's always, so CDISC is for clinical patient level data. For the rest, it's a lot. We rely a lot on document. There is a few structured data in what we are doing today. So, for example, XCVMPD, the drug vigilance database from Europe, we have to submit after getting the authorization or after getting the variation approved, some data to authorities. And for SMS, the same. Before to use a substance in a product for a CTA, for example, we must register it, but it's very, very basic information. It's basically a name, just a name. So when you have a protein with a long sequence of amino acid and so on, you can imagine that the name may not be enough to describe the protein properly. On the FDA side, it's also a similar process. So SPL has to be submitted 14 days after application approval. And for GSRS, the substance can be created from scientific information, but it's quite independent from any regulatory process. We can request any substance when we want, but it's just that we need this Unicode before SPL submission. So as we see on the bottom line of the very, very high level development, it's EU-based with I took EU process bits. There is not so much structured data. So what will be the consequence of IDMP implementation here? Next slide, please. Is that we are going to switch to a much more structured-based exchange. And we have today, with the DADI project in the EMA, all this approach of putting data into the submission, into the variation. So it's part of the process of the variation submission process to negotiate the data as well as the content of the document. So it's really opposed to what we see in XCVMPD today is after you get everything is done, every document approval is done, then we submit the data. It's completely reversing the process. We have to manage in parallel the data exactly aligned with the document and go to the negotiation with authority like that. So the consequences is that we need to manage the data lifecycle in parallel of the document, and it's mean that when you submit a variation for a specific product, you have a set of approved data and a set of data which are currently in negotiation. So that means we have two sets of data for the same product to manage at the same time. That's for the submission. Then if we think from a scientific standpoint, it's also sometimes a difference. Let's imagine we have a shelf life of a vaccine. We know that from stability studies that it's stable for 10 years, let's say. The antigens are stable for 10 years. But what we have requested, what is authorized is that, let's imagine 2 years. And today we are sending a variation for 3 years of a new shelf life for 3 years. So that's this kind of all these data points to qualify properly each of these data points to make sure that we are not mixing up of this is creating an additional complexity within the industry. In the past, it was much more document-based. Now it will be data-based. And also another complexity, which is already existing today. I'm not saying that it's completely new. It's already existing in document, is the parallel desynchronized submission. So we have a specific submission in a country with a specific shelf life and a different one in another country with different shelf life. So that means we need to manage all these different data in parallel, all together in this broader context. So as we see here on the timeline below, we have more green boxes. Also, I forget to mention CTs with the submission of structured data for clinical trial. Even if with the CTA form, there is already the structured data in the XML document, but still it represents an additional complexity to manage. So next slide, please, Christian. And then, so having more data structure is also that we have more data object representing these different elements, these different concepts to represent. And this more data object means also more identifiers for this data object. So on the EMA side, so I take the EMA example here, we'll have this PMS stable ID. So this product will have this ID for all its life. MA number, this is already existing. MPID, so tomorrow PRD code will be superseded by MPID. there are the identifiers for the pack, the PHP ID, the substance ID, and on top of that, if we have global IDs, we'll have also the global PHP ID from UMC, the global substance ID from UMC, and potentially there is a project, a proposal in the TC215 to have a global medicinal product ID. So all this ID needs to be managed properly. So it's really a complexification for us of the process, data in parallel of document, and to be managed all through the life cycle. This means that data quality need to be at the best. We cannot, let's say, identify the wrong pack, the wrong submission will be done. I will not update the right pack, for example. But also it improves transparency, because if we can immediately say, okay, this is this specific product, this specific PHP ID, it's much more faster, much more straightforward. Say, okay, yes, I know exactly what I want. I know exactly which product I'm referring to. And it's also better in terms of life cycle management, pharmacovigilance on product, because we can immediately say, okay, that's this specific product we want to identify in this specific process. So all this is, I think, really critical. There is a lot of benefits around this, additional data. So on the product aspect more broadly, but on the specific of the substance, it's the same. So next slide, Christian. So now that I explained the principles, what it's been in terms of implementation. So as I already mentioned that a little bit, that it's additional data point, additional capabilities, system process capabilities, additional controls, training for people. That's challenges, but there are also a lot of benefits. Many times in companies, and I think my colleagues from the pharma world will recognize that we speak about a product, and we don't always care to say, okay, it's a product or a salt of that product, this specific salt or this other salt of that product. We just mentioned the active moiety, this active substance that is in this product, without really referring to the exact substance that is indeed the active ingredient in the product. And having a very straightforward, very clear, scientifically driven identification of substance will be a real benefit. Here I have, sorry. I have an example for a vaccine. We have, for example, a polysaccharide that is conjugated, that is adjuvanted. So these are three different substances. It was nicely shown by Annet in the slide with all this hierarchy of substances. So we need to be clear on what we are speaking exactly about. And sometimes, if we take a specific active or a specific active ingredient from one vaccine to the other, it will be the same the pathogen that we want to tackle, same for the two vaccines for the same pathogen, but the adjuvantation will be different. If I take for authorized product, our Hep B vaccine, Engerix, is adjuvanted on aluminum. Our Fendrix vaccine is adjuvanted on a specific adjuvant system. And these two, because we have different target population and so on. So saying just Hep B vaccine is not enough. We need to say exactly which Hep B vaccine we are referring to. Also, when we have a specific polysaccharide like this, for example, if we have just the one that is produced naturally when we culture the pathogen, that's good. But sometimes we also have some treatment of this polysaccharide. Let's say we have a mechanically sized polysaccharide. So the average molecular weight of the polysaccharide will be different. So we have to differentiate, to distinguish two different substances, because there are consequences on the product, because more the polysaccharide are long, more the vaccine is viscous, more it's painful, and also there can be some consequences on the immunogenicity of the vaccine to mechanically size the polysaccharide. So the other benefit is that we have immediate alignment everywhere. So if we have one set of data clearly identified with the same global substance ID, same PHP ID in Europe, in US, we can imagine that the pharmacovigilance is much faster. And when you have a very low level signal in a country alone, it's not meaningful. In another country alone, it's not meaningful. But when we put all this together, maybe this low-level PV signals start to make sense. So that is kind of stuff that is helping here. And helping us also in reporting of the adverse event. In terms of regulatory perspective, it's also more straightforward. If we have already the substance, the product, well structured, clear, okay, we know exactly what we are speaking about, it's clear this product, this substance, we gain in efficiency. And you know that in pharma industry, we want to have the product on the market, I mean, as soon as possible. I mean, we are not a non-profit organization, so at some point we want to sell the vaccine. And in terms of when there is a pandemic like today with COVID, we see that the quickness, I mean, the fact that we are quickly on the market, the fact that we have quickly a vaccine available is something critical from a global health perspective. Also, in terms of transparency of the information, when we have a clearly structured information, okay, we can immediately analyze it. It's easier, faster, also to compare, for example, the different vaccines. So that's easier. Instead of going in document to have to deep dive to analyze all these pages, it's a more complex way of working. So all this represent, of course, an improvement. And something also we need to think is, on a pharma perspective, when we follow up pharmacovigilance of paracetamol, we focus on the substance because it's meaningful to make the follow-up on the substance paracetamol, regardless where it's coming from. It's coming from supplier A, B, or C, doesn't matter. What is a bit complex in vaccine is the Hep A vaccine from GSK. Is not the Hep A vaccine from Sanofi, and it's not the Hep A vaccine of another supplier. We are in a biological world where a substance, I mean, biosimilar are not generic of each other's, if we can say like that. We could think that this different vaccine with the same antigen could be seen as kind of biosimilars, but in fact, it's just different substances, different. It may come from different strains, different proteins that are expressed in the process. Even if they are slightly the same, they are still different. So it's different substances. So that's also a critical difference for vaccine, is that we need to be very clear on the substance and to identify the product based on that substance. So the PHP ID will be different from these different products. So next slide, please. So yes, so the challenge I said, or data point business, also the complexity is to make sure that business and users, our CMC colleagues in the regulatory function and so on, get all this complexity. And we are also shifting the knowledge from regulatory to the scientific people in the development function that has really the knowledge of the substances. So as I said, capability, so time to learn, we need additional controls, a change in mindset. We have today a mix of system and document-based of all this information. We need now to put all this in the science system managed by scientists, by a scientist function, to support a highly precise antigen information. That's also something critical. What is also another complexity, not really linked to IDMP in itself, but linked to the context, is we have already sometimes more than one system to manage substance, more than one standard in the system to manage substance. So IDMP can be seen, and is seen by some of my colleagues, like, okay, that's one more representation of the substance. The fact also that it's very transversal for us to implement this such a process to collect substance information, because the antigens start to be developed very early on in the early R&D. So we have a first description of what will be the antigen very early on. Then it's transferred to development, which will elucidate additional information, additional characteristics on the antigen. So all you see, it's a continuum from early R&D until the file. It's a continuum where we have to to be able to enrich the information, enrich the substance information. Still, at several point in time, we have to submit some information because we have the very first submission will be scientific advice, where we have to start to declare a substance. So, but we may not have the complete, at least for vaccine. I know it's different for chemicals, stuff like that, but for vaccine, sometimes we have just a high level description, high level knowledge on the substance. It's not always completely described from the beginning of the R&D. So all this enrichment process is also something that is important. So as I said, also the scientific aspect, this I already tackled it from a scientific description. What we are used to see in our development and commercial function is a focus on material. So this concept of material is not, I mean, we don't, when we produce a vaccine, we don't really care about the specific substance, protein, isolated, alone, very pure, and so on. What we get from the previous step of the development is a product, is a substance, sorry, in a medium with some level of impurity, something like that. I mean, it's not a pure, it's not a pure antigen lyophilized, for example. At least not often. So that's also something which is a change in mindset. Is we are shifting from a material-based, lot batch-based approach to a scientific-based approach, and people are not used to really manage oftenly this dimension of the product. Yes, they do it, but it's more okay. Once a while, they have to report on a specific aspect of the substance, and then it's done. Also, a last point is from a pharma industry perspective, IDMP is still seen as a very regulatory driven problem, if I can say problem. So it's really a substance standard that is used to submit information to authorities. And so we have to develop the knowledge down the line in the development function, because they will be instrumental, especially for substance, maybe a little bit less for product, but especially for substance, to provide the scientific detail on the substance. Next slide, please. So then, all this, what does it mean in terms of pharmacovigilance? Of course, pharmacovigilance in industry will leverage on the PHP ID and global substance ID to identify the product in the case safety report, in the individual case safety report in the database. There is some challenges for us, as Florence wrote in the chat. Maybe I will answer later. To make sure that we can identify the product, indeed identify it, and then being able to identify across the different jurisdiction. And the same for the global substance ID. If we have an HEPA vaccine, okay, which HEPA, which HEPA antigen, the GSK, Sanofi ones, another one. So that's another challenge. Also, what is interesting for the PHP ID is that this contains the pharmaceutical dose form. And the dose form characteristics, which are where we are shifting to with the revision of the 11239, if I remember well, ISO, on the dose form, will help to improve the granularity of the reporting. What is interesting for vaccine also with the global substance ID, I said earlier that sometimes we have the same substance in different products with different formulation, with one adjuvant, another adjuvant, or we have even combined. When I said Hep B vaccine, we have Hep B with aluminium, Hep B with adjuvant system, Hep B with Hep A for combined vaccination, Hep B in pediatric vaccines. So if we want to analyze Hep B antigen consequences, then it's transversal. So the global precise substance ID definition will also help to have this transversal analysis through the portfolio. And the same for antigen. We got in the past question from authorities, what about all your studies regardless the adjuvant on this specific antigen? So that's important. So it was said already by Olof that sometimes the reports are quite vague for vaccine. So the missing trademark that I said have vaccine, Also, sometimes the population, we know that it's an Engerix vaccine, but is this Engerix for adult or pediatric? So it's two different PHP ID because it's two different strains. So that's a challenge. Also, something which is sometimes challenging is that the PHP is not covering the adjuvant. The PHP ID does not take into account the adjuvant. So, I mean, the excipient, sorry. So the adjuvants are not seen because they are seen as excipients in the PHP ID. And also sometimes we have some adjuvant, some excipient that may have an impact, and we need to be able to analyze across different products the excipient. So that's also a limitation of the PHP ID. And so I already mentioned the fact that the biological substances are not identical, and that's a challenge. Next slide, please. So now, all this, how industry can contribute to this global substance ID and PHP ID setup. Next slide, please. So that's where I think industry has a pivotal role, because industry is really the originator, owner of the information. We do develop. I mean, we create the information by developing a substance, and of course we are expert on this substance information. Also, so that's why I believe the industry need to be involved heavily in the exchange and the creation and sharing of substance information with regulatory authorities. It's great that regulatory authorities can build a database from the file and so on, but there is, I think, limitation also that where we can help by having our specific perspective, more specific information of substance. Also, so as I said earlier, the vaccine development is sometimes complex. It's sometimes complex to characterize everything, and we do it all through the life cycle. And so we start with the basic description in the early R&D, and more we go to close to the file, more we have the information around the substance, and that's that's why we really need to have this minimum substance field to say, okay, that's really the very basic minimum number of fields that we can use to identify at least high level of substance. Okay, down the line there will be more information. Maybe down the line we will a little bit fine tune the substance information, but at least we have this very minimal set, which is not scientifically sound, which is not preventing to be ISO compliant when we file, but at least for this very early phase, very first scientific advice, then we have something which is start to be scientifically sound. This means that down the line, we need to be able to manage the life cycle. So how to create new substance and enrich them, create parent-child relationship. For example, let's imagine we have a protein. We start very early on, first scientific advice, we say, okay, that's our nice protein and everything, but we haven't elucidated the disulfide bond in the protein. Down the line, we realize that in fact there is several isomers. So in fact, the one protein we feel we have at the beginning, in fact, it's not one, it's 3 or 4 different substances, 4 different proteins. So they are not isolated, each of them are not isolated alone. They are all mixed in the product, but we still need to describe, to be scientifically sound in the GSRS system. We still need to define these 3 proteins, these 4 proteins, and then we need to be able to manage this parent-child relationship between the proteins. So all this aspect of having scientifically sound, something which we can develop over time, all this is really critical. Next slide, please. So here you see that I steal a slide from Annet to highlight in my presentation, but I think it's really critical. I mean, this slide is really important, and it's also connecting to the very first schema that Olof was sharing in his presentation. Is we have these different databases, so this schema with the different regulatory authorities database on the left. In fact, there was this industry database alone on top. In fact, we should connect the regulatory database, the regulatory authority database together. We really have the information, and we can act as a broker, if I can say, between regulatory authorities to exchange the same information, and this information should hold the public signature, so each one can check that we have the same set of information. And that's the proof of concept we are currently developing with the ESRS team. with the 3 companies Annet mentioned earlier. Next slide, please. So in terms of how to make this exchange possible, we should be able to leverage on SRS system, and that's where industry may install an SRS instance internally to be able to structure and to export JSON file. So we have the same system everywhere, same data model, same export standard. I mean, we could imagine that in the future, long term, we just generate a JSON from our legacy systems, but let's, at least for the proof of concept, like in GSK, we install an SRS instance. We export that to the SVG, we share this information with them, and then the expert from the substance validation group will say, yes, okay, we agree, that's a new substance, that's structured in the way with the business rule and so on, and it's confirmed, we give you the substance ID, and then we are there. Or we can discuss, okay, there is disagreement, we don't agree the way you structured it and that and so on. We can imagine that, and then at the end, we got feedback from the authority that this substance, the JSON file we sent, is coming back to us, validated with the ESRS code, the UTCT code attached to it. So to make this proof of concept, we need to define the rules, the process also, this is a high-level process. This will be also the occasion to make sure that the business rules that has been already defined by substance validation group are working everywhere. In the multiple case, we'll be able to handle. And then you see the hash process. This is to have a public signature. So to have a subset of the data, a subset of the data that represent the substance, which are definitional for the substance. And we have a hash result hash calculation, and this will be a public signature. And then what it means for the next authority is we send the same JSON to the other regulatory authorities with the hash calculation. This other authority can indeed take this data, rehash them, and ensure that both authorities have the same hash result and so the same substance. So without talking to each other, the 2 authorities will know that they will have the same set of data. So FDA, EMA are not allowed to exchange confidential information, but in this way, they know that they have the same confidential information in their database without saying it, just because industry will share the same file and the hash will be public. And then for UMC, it's easier. We make an extract from this substance public information, FDA, EMA, or even industry, send to UMC with the substance version, the substance hash. And so UMC doesn't need to know what is making the substance unique, even if they have another substance which is identical from a public perspective, public data perspective to the first one, they know that it's a different one because it has been validated by authorities. The hash is different, and so they can confidentially say, okay, it's a different substance, even if it looks from the public data the same substance. On the data aspect, it's important also to make sure that these data are not modified. So when they are validated by authority industry, then they are not modifiable anymore, and they cannot be enriched, at least for what is definitional. You can add an additional CAS number, you can add an additional publication linked to that, but not a new data. So, final remarks. As I said, it's a paradigm shift for industry to go for this much more data-driven process. Definitely, it will help for pharmacovigilance. I don't feel that industry will have a pivotal role because, I mean, pharmacovigilance is mostly held by regulatory authorities, by public institutions. But of course, we have a facilitating role to make sure that this case is well identified. The definition of the global substance ID and PHP ID, I strongly believe that we need to involve the data owner, so the pharmaceutical industry, because we are experts on the content, on the data themselves. We know our data. And and obviously this will be a benefit for us to have one definition, one global definition of the substance and the product. Thank you very much. Now, I think we are switching to the question, Christian.
Yes, thank you, Jean-Gonzague, and thank you to all the speakers. We are going to have a rather short debate because the time has flown quite quickly. Thank you for the audience who has asked questions. I'm going to select a couple of these questions because the time is, as I said, a bit constrained. If I select Olof, I wanted to answer that question from France Williams. EU also requested no longer send ICSR mentioning only flu vaccine, influenza vaccine, etc. Would you be able to answer that shortly?
Yes, and I would give my view on it because it's since I'm not working for a regulatory like the EU, so I just know on the global perspective what we actually get in on the ICSR, and that's where we see the need to group them, maybe on the influenza vaccine, for instance. But of course, the more information we have, and I understand the regulatory perspective there, that you want to have the detail, because otherwise it's difficult to do good analysis on just flu vaccines. But we also know that that's the reality, that we get a lot of those reports, at least on the global scale. So I don't think I can answer it fully, because I think some from a regulatory perspective, or maybe you have from the industry even more, Jean-Gonzague.
Yes, it's a little bit what I mentioned during the speech, is that we should be able to have a kind of hierarchical PHPID at a higher level. So let's say we have a kind of abstract PHPID at a virus level. So it will be, okay, you have your PHPID virus level. So if you don't have the name, the trademark of your HPA vaccine, you put at that level. And then if you know the exact strain, exact product, then you put at the exact PHPID. But I mean, that's mean in some ways, UMC, you should generate a series of abstract PHPIDs based on each virus strain, let's say, where we could put this kind of ICSR report that are not well defined, in fact. That's something, that's really a request from the industry. If we want to really put an PHPID on each of these ICSRs, we need to find a way to generate fake, or abstract PHPIDs.
Thank you, Jean-Gonzague. I would like to shift to another type of a question. We have actually a Swiss colleague asking the question, do standards like SNOMED play a role for substance identification? And I would like to extend that question also to INN. What is the role of INN in the substance identification? And because we have the chance to have Rafaella Balocco with us, maybe Rafaella, can you say something? Because we had already a webinar on this situation. How do you see the relation between INN and the IDMP substance? And after that, I would like eventually Annet to give the other perspective. Rafaella?
Well, I think that in a global world, you need a global way to a global language, if I may say so. Databases are nice, but people speak using languages and words. And to me, maybe I don't want to be simplifying the situation that is always much more complex, but I think that to be able to call things by their name is super important for everything, and even more in the medical field. So INN historically has been born to be able to have a name for a substance. I'm not talking about a product, which is very different, a substance. And until very recently, we were not dealing with vaccines because the vaccines were not sufficiently well defined. And we do wish to name substances that then we are capable to describe. So I'm Rafaella, this is my name, because I am this person, I was born by these parents, and I have these characteristics. Same for a substance. You are tosinemeran because you are the Pfizer BioNTech vaccines that has been created through this process, and it can be described in this manner. And now we can name, not all, but the majority, I would say, of the modern or more modern vaccines, the RNA, the DNA, the recombinant protein units, the proteins, the peptides, the viral vectors. In reality, we could have easily named also the AstraZeneca, and if we didn't, it's because AstraZeneca never wanted to have an INN, or I mean, for their own reasons. I don't want to enter into details now, but and then, of course, there are the traditional vaccines that are not so well defined, and the scope of this is out of the scope of INN. Still, we have another group in WHO, which is the Expert Committee on Biological Standardizations, capable to name those vaccines. I don't know if I answer, but to me, when it was said that vaccines are like biosimilars, well, obviously all are biologicals, because biosimilarity for me is really a regulatory component, is not a scientific component. So if you want to remain, of course we are talking to regulators, but if we remain in science, all biologicals are by definitions grown in living cells. They cannot be reproduced so exactly, but this is for now. We don't know for the future. We cannot predict in 10 years from now, in 15 years from now, maybe somebody else will be able to reproduce exactly tocilizumab, although it's not BioNTech. I don't know. I am not a virologist, but at the very least, we will need to be able to say that this is tocilizumab. And even more now that new modifications of these vaccines are coming, because maybe I'm not saying a secret, you know very well that the Omicron one will come very soon to the market, and we are naming this. And maybe, I don't know, I am just speculating, and it's a pure speculation. They will mix the two. So one shot, but with the, let's say, the traditional tocilizumab and the Omicron tocilizumab, and they will be mixed, and you will only have one shot possible. And then, and if we can imagine that there will be many more of this, then you will need a simple way to call, or at least to know what there is inside. So for me, it is not all, and certainly not the only component in a pharmacovigilance system, because we're talking about pharmacovigilance for vaccines, but a very important one. And of course, because we are in a modern world, and we do not only rely on our brain and on our capability to talk, we also need databases, and databases can incorporate much more, and is where all what we have been discussing today comes in. So this is a component, a very important one, a very important part of pharmacovigilance, and is a part of a more comprehensive picture. Over. I don't know if I could answer.
Thank you. Well, it is a complicated question, a simple question with a complicated implied response. I said eventually Annet can say something, because the question is more linked to the interaction between this and that standard, so that standard being IDMP substances. Well, I would also have asked Jane Miller, who is on the call, but first perhaps Annet. Just your perspective regarding the relationship between INN and SNOMED.
Yeah, I'm not sure if I can answer. The only thing I can answer is that the INN itself is for the substance validation group a key trigger to review the substance fully, and to make sure that all the information that we need is in the substance. And we are at this moment not taking into account the SNOMED as such, as far as I know. Maybe we could. Is it? I'm not sure if SNOMED is also on substance level. I sort of related that to products.
Yes, I think so. Maybe Jane Miller can say a word on that. You have the microphone, Jane.
Thank you. Yes, it's Jane Miller. I'm not the substance expert, but I can say yes, we do have substances, and I think in the use cases you're talking about, obviously, if people have a SNOMED drug base and are using the drug model, then they may well be using the substances. And therefore, the relationship between the substances used at the clinical end, and because obviously we need substances for adverse events, and not just related to drug causes, but to other causes. So inhalations and things like that. So if I take that, and accidents and things. So I think that what we're trying to do with substance is make sure we're aligned where possible with both the clinical end, but also the requirements for regulation. So we welcome, as always, and Malin knows this herself and others within UNICOM, any direct requirements that start to emerge as we move into the world of IDMP data and regulations starting to use that in the different categories. Yes, we have, and we have been watching all the different developments. But the question is, what do we align with when you've got something happening in Europe, something happening in Asia, something happening in the US? And yet we have got to align for clinical care and all the different incidences where substances may be related. Thank you.
Thank you, Jane. You know, the time is flying out, and unfortunately, we have to limit ourselves. That's a bit of shame. There are some pending questions which are really interesting, but we will try to answer them in a written way afterwards. So unfortunately, I have already to stop this debate by thanking the speaker and panelists, but also the participants. Today, we have had at the top something like 120 participants, if I was well aware, including on YouTube, by the way. I invite you to respond to our questionnaire. You find the link in the chat box here, but you will also receive that in an email, as I said, tomorrow. Please respond. It is extremely important for us to have enough responses to this questionnaire, which is very quickly done, by the way. Our next Committee of Expertise will take place on Friday, the 4th of February, so very soon, and it will be around PHPID production and all what that implies on the clinical side, and of course also on the regulatory side. I think it will be at least as exciting as it was today. Exciting in the sense that I suppose many learned a lot, and that's the purpose of a Committee of Expertise. That said, thank you for having joined. Thank you for your patience also, and I wish you good health and a good weekend, and hope to meet you in about 2 weeks from now on the 4th of February. Goodbye.
Thank you. Bye-bye.
Bye-bye. Bye-bye, everyone. Thank you.